So I'd just like to start out my talk by thanking Bynano for their opportunity to share our work with you, and also to say what a thrill it is to sort of share the podium, if you like, with Crystal Depian, who's been a fantastic collaborator for us on this work. And so today I'm talking about these new class of repeat expansion disorders, which are AT-rich pentamers, and specifically in the cause of familial adult myoclonic epilepsy. Just as a brief introduction, I'm from Adelaide, which is in South Australia. And if you have any questions about this talk, please feel free to contact. So today we're really talking about repeat expansion disorders, and there are actually now over 50 Mendelian versions of repeat expansion disorders. And if we think about these disorders in the context of a typical gene model, a coding gene model, these repeats can expand and cause pathology from anywhere within this model. So promoter, 5 or 3' UTR, coding, or intronic regions. These repeat expansion disorders are characterized by the changing in the size of these repeats from smaller to larger, and that can occur both somatically and across the germline. And the mechanisms behind this are believed to be tied to DNA repair, replication, or recombination. As a result of this unusual dynamic phenomenon, we see in these families the phenomena of anticipation, where disease onset is typically earlier and symptom severity can sometimes be more severe in successive generations of a family. And this is typically correlated with an increase in the size of the repeat. In terms of how these repeats cause disease, it can depend to some extent on where the repeat occurs. So if it's in the coding region, for example, you can get a novel protein function. But even for the non-coding repeats, you can get altered levels of gene regulation. The repeats can be transcribed, and that RNA can form toxic foci, so the RNA binding proteins can be sequestered in the cell or cause aberrant splicing. And then also the repeats themselves can be translated by a phenomenon known as repeat-associated non-ATG translation to form these smaller peptides corresponding to the repeat sequence, and they can be toxic as well. So the disorder that we're interested in, and I'll tell you, we've been looking at this disorder for a long time, is familial adult myoclonic epilepsy. It's characterized by typically a cortical myoclonus, which manifests as a hand tremor predominantly, and that can get worse with exertion. In at least 30% of cases, many individuals go on to also generalize tonic-clonic seizures or myoclonic seizures, basically epilepsy. As I said, it's an adult onset disorder, an autosomal dominant disorder, and the onset of the tremor typically occurs in the 2nd to 3rd decade of life. And the disease is mildly, but not very highly progressive. We know of 6 genes that are linked to FAME, and today I'll mainly be talking about STAR-D7, and I know Crystal will be talking about the March6 gene causing FAME3. And what's peculiar about all of these genes, even though they're completely functionally unrelated, is that they all have an intronic TTTA, which is a reference repeat sequence, typically quite small, maybe 5 to 7 copy numbers, and an inserted TTTCA repeat expansion. And these expand to many hundreds of copies that all are associated with these 6 genes and all cause FAME. So I'm going to tell you a bit about our story and how Bynano has helped us to learn some new things about this disorder. And a lot of our story is linked to this large Australian, New Zealand pedigree collected by our collaborators Ingrid, Doug and Lynette in Melbourne and New Zealand. And a lot of the linkage work which I'm presenting on this slide was performed by Melanie Barlow's group. So here we started out our story in around 2012, and obviously collection of this large family started well before then. And we can see in this original work that the linkage was narrowed down to 13.3 megabase region, spanning the centromere on chromosome 2. We formed an international collaboration with individuals in Europe and the US. and obviously the Australian groups. And we formed an identity by descent mapping. So we looked at whether haplotypes were shared across multiple FAME families, or about 20 odd different families affected by FAME. And we were able to use that to show that there were several founders, and that these were and helped us narrow the linkage interval down by another 3 megabases. There's still quite a lot of genes in this region. And then finally, just before this time last year, we were able to use various sequencing techniques to identify the repeat expansion which causes FAME2. And that was quite a relief for many of us. So what I'd like to do is just briefly show you some data from that paper. So this is what the FAME repeat expansion looks like on short read whole genome sequencing data. So this is probably what most people would use to look at this kind of situation where you have a large unresolved family. And you can see this is an IGV shot. So we have everything that's grey matches to the reference, and then these reads which are in green and red, or green, orange and red, are indicating reads which are not mapping to the reference, and that's because they contain this repeated sequence. You can use short read sequencing techniques to measure the sizes of these, but there's some problems with that method in that it doesn't quite get the right size. So I asked the question for the course purposes of this talk, what if we had used Bynano instead of short read sequencing and whole genome sequencing in 2016? And so in collaboration with Jill Lay and Alex Hasty at Bynano, and we're very thankful for their cooperation with this work, we performed Bynano assemblies on 4 individuals from this large Australian New Zealand family. And here you can see the typical mapping data. And of course, this is for chromosome 2. And I've just marked the unaffected allele in this darker blue, and the affected allele in this lighter blue, and the green is the reference map from Bynano. So on de novo assembly, we see that every one of these individuals has a repeat expansion on chromosome 2. And in fact, if we overlap them all across in something like the UCSC genome browser, we can see they map directly into the first intron of star D7 and directly where this AAT repeat expansion occurs endogenously in the genome. So basically, in one experiment, we would have found exactly where this mutation was, gone from a 10.3 megabase linkage interval right down to a couple of kb, and found this expansion very rapidly. In the last few minutes, I'd just like to quickly go through some of the data that we're moving forward with on this project. So we have expansions, now a method of measuring the repeat expansion across multiple individuals using long-range PCR. And here we can see in successive generations of this Australian family that we do see increases in size of this repeat through the germline. And that this is correlated with work that we published showing that there is clinical anticipation in this family where disease onset is becoming earlier in those same 3 generations. We've looked also at the inheritance of the repeat, maternally or paternally, and we don't see any germline specific effect from the parent of origin. In terms of looking across the population more broadly, so multiple individuals affected with FAME, we've measured, it's been measured in FAME1, and Crystal, I'm sure we'll talk about FAME3, that the size of the repeat is anti-correlated with the onset of the myoclonus or the epilepsy. So basically, the longer your repeat is, the much earlier the disorder will onset. So just to briefly summarize our FAME2 story. It's caused by repeat expansions of TTTA, which is reference, and TTTCA, which is non-reference. We've screened that in 158 clinically affected individuals over 22 different families, and they all tested positive for this repeat in star D7. We have anticipation, which is observed across all forms of FAME that we've looked at to date. There is both germline, and I'm sure Crystal will talk about the somatic repeat instability in FAME. And the repeat length and copy number is anti-correlated with the ages of symptom onset, particularly the TTTCA repeat content. And of course, Bynano optical mapping is a very efficient way to identify novel insertions, and certainly a technology that you should consider in some of these unresolved families. And then I'll just quickly flash up our slide, thanking a lot of the people that have been involved in this wonderful project. Thank you very much.